So welcome everyone. Thanks for joining the webinar. Today we're talking about the 7th annual Invasive Species Mapping Challenge for New York. We're so excited to run this again this year. We've gotten a lot of great data over the past few years, a lot of great participation, and we really like to continue that. So I'll give some background on the challenge and IMAP, and then we'll also hear from some experts today. So we have Maria Moscali from the DEC to talk about beech leaf disease, and we also have Anna Stopson from Yale to talk about jumping worm. So kind of the goal today is to learn how you can participate in our mapping challenge and learn what it's all about. So we'll talk about how to identify the focal species. So there's pictures of the 6 different species on the right-hand side, all from IMAP records. And then hopefully you'll all also be on your way to reporting observations to IMAP of these species. So those are kind of the 2 things today. And I should start by introducing myself as well. So I'm Mitchell O'Neill. I work with the New York Natural Heritage Program. Our main focus is rare species and biodiversity conservation. That's the main focus of our organization. But since invasive species are one of the biggest threats to biodiversity in New York State and globally, we also administer the invasive species database for the state. So that's why we administer iMapInvasives. So I work in the invasives section of the Natural Heritage Program. And just so everyone's on the same page of what invasive species are, so these are species of plants, animals, insects, or pathogens that are 2 things. So they have to be non-native, meaning they come from somewhere outside of New York. And they also have to be negatively impactful. So some sort of harm to the environment, the economy, or human health. A lot of invasive species check all of those boxes. An example on the right-hand side is a picture of spotted lanternfly, which is an invasive insect, and it's swarming a grapevine in this photo. They suck the sap out of woody plants and there's really a lot of concern for the grape industry and forest industry and all sorts of other things, really far-reaching impacts. That species is a very high priority species that's making its way into New York. It's not in most of the state yet, but it's making its way. So invasive species, they cause all these problems and they're very difficult to manage. And our strategies for managing them really varies from species to species. One of the things it varies based on is how abundant a species is. So species that aren't here yet, or maybe they're just found in a couple places, we really want to focus our resources on eradicating them or preventing them from spreading. But as they become more abundant, that becomes less possible, and we have to focus more on containing them or prioritizing where we do our management. But really to figure out what we're going to do about any given invasive species at a regional or statewide level, we really need to understand how widespread it is. So where to, like what the distribution is across the state. Is it all across the state? Is it just in one part? And also what specific locations it's at. So if we want to manage a plant, for example, we need to know where it is specifically. And that's where iMapInvasives comes in. So we're the centralized invasive species database for New York State to support state agencies, PRISMs, other partners working on invasive species issues. You can use IMAP to report observations, to view data, to view invasive species distributions, to track control efforts, and more. And I just said PRISMs. So for anyone who's not aware, these are the partnerships for regional invasive species management. I really encourage everyone to check which PRISM they're located in and check out their websites. You can find information on all of them at this link here. And they do a lot of great work, so I encourage you to check them out. And so IMAP is this big database. Where does the data come from? So at first, it was really important to gather data sets from partners and get them all in one place. But now we're in this paradigm where the most important data source is data entered by community scientists and professionals into iMapInvasives in real time. So we really thank all of our partners and everyone on this call for helping us keep our database up to date. We really rely on you to help us keep track of how invasive species are shifting, appearing in new places, all that sort of stuff. So what is the mapping challenge? So this started back in 2016, and it really had two main goals. One was to fill data gaps in iMapInvasives for certain species. In the beginning, it was water chestnut and tree of heaven, and then we eventually added jumping worm, and then added more species from there. And then the other goal was to just grow our user group so that we have a bigger base of people monitoring invasive species in New York. So it really takes everyone to monitor invasive species. So we have professionals doing surveys, but we also have volunteer community scientists out there looking for invasive species and reporting them when they see them. So community scientists are very critical to mapping invasive species. And so if you're looking for, I hope it sounds interesting to participate just from that, but if you want some more incentive, we do also have some fun gamification with these challenges. So there's prizes and trophies. So the top community scientist or volunteer reporter of presence and not detected for the length of the challenge, which is starting tomorrow through the month of July. For any of these four species or species groups will get a prize. So the top reporter of tree of heaven will get a prize, top reporter of jumping worm, top reporter of beech leaf disease nematode, and then the top reporter of the combined total of the three different aquatic species we have this year. And to give you some ideas of past years' prizes, sometimes invasive species guides, Conservationist magazine, coasters, spotted lanternfly scraper cards. We did a smaller challenge for New York Invasive Species Awareness Week a few weeks ago, and for that, people got 3D spotted lanternfly puzzles and boot brushes and temporary tattoos. So lots of fun little things to look forward to if you end up winning. And so those are up for grabs for individual participants. But there's also trophies up for grabs for the prisms. So all the volunteers doing their reports within each prism and staff as well. We sum those all up at the prism level, and then the top reporting the prism with the most reports for these four we'll get a trophy. So there's 4 trophies up for grabs, one for each of these species, and then one for the combined totals of the 3 aquatic species. And I do want to reiterate that that does include presence and not detected records. So even if you go out and don't find it, that's really important to document and can help your prism get a trophy. So the challenge kind of centers on a list of focal species. So these are the list of species. We've got 3 individual terrestrial species and then this group of 3 aquatic plants. I won't go through the details of all of them right now since we'll hear a lot of that from presentations later. But essentially, most of them have been added onto this list for some combination of reasons, including they have impacts that concern our partners like agencies and prisms, and there's some sort of data gap. Like there's regions where we don't have enough data, or maybe it's an emerging species in some region. Those are kind of why we put those on our target watch list for this challenge. And just to, I said data gap, so what does that actually look like? So here's the map of Tree of Heaven in New York State from the IMAP data. So the green polygons, those are towns that have Tree of Heaven reports in them. And then the yellow dots are showing where people have searched for Tree of Heaven, but not found it. So that's always really good news when you look for an invasive species and it's not there. But there are some gaps. So there are some places where we're not really sure if the species really isn't there or if it just hasn't been checked for yet. And so the other piece, they're all species that are of some sort of concern to our partners. Just an example for Tree of Heaven, it's a species from Asia. It grows in disturbed areas, both urban and natural areas, but it always needs some sort of light gap for it to grow. But it can even grow like right in urban areas, right out of the concrete sometimes. And the impacts, one is that it crowds out native species because it grows so dense and shades out other plants and actually puts out compounds into the soil that make it harder for other plants to grow. And the big thing with Tree of Heaven is that it's actually the preferred host plant for the spotted lanternfly that I was mentioning earlier, which is one of the highest priority invasives in New York State. And since we're not going to do a big training on Tree of Heaven, I just wanted to make these 3 notes on the identification so people are familiar with it. It's a tree. It can grow quite tall, over 50 feet tall, but it's often seen as like a smaller tree. And one of the characteristics of it are these big pinnately compound leaves. So this whole thing right here is a single leaf, and the little things around it are all the leaflets. So that's a very distinctive characteristic. There are other plants that look a lot like this, like sumac or black walnut. But the thing to look for with Tree of Heaven is that it has these smooth leaf margins, so there's no jagged teeth, except at the base of the leaves. There's these little round teeth, these little bumps at the base of the leaflets, and they have a little glandular dot on the back. And one other feature is that they have a smooth cantaloupe skin resembling bark. And so that's kind of a brief overview of what the challenge is. And so now we'll get into more detail about some of these species. So first we'll hear from Maria Moscali from the DEC about beech leaf disease.
Hi everyone, I'm Maria Moscali here from DEC Forest Health. Today I'm going to give you a brief history of beech leaf disease and show you how to identify it and its lookalikes. I have a lot of slides, so I'm going to talk really fast. Sorry about my dog. That did not go forward. The controls don't seem to be working.
I'll try it real quick, and if it doesn't work, then we'll just...
I can just say next too. I'm okay with that.
Yeah. Can you try one more time?
Yep. Okay, that is not working.
Okay, no problem. I'll just...
Okay, so I'll just say next.
Yeah.
Okay, so next.
Okay. And also a quick reminder to all the participants, feel free to enter your questions and thoughts into the Q&A. And there's also the chat box if you can't find the Q&A. And I'll hand it back over to Maria.
Okay, so the importance of American beech. American beech trees are an important component of the northern hardwood forest type, which makes up more than half of the forested land in New York. Wildlife such as black bears, squirrels, mice, turkeys, and many others rely on beech nuts as a main source of energy due to their high protein and fat content. And then predators such as owls, coyotes, and others are also affected by beech mass indirectly when their small mammal food source dwindles. Beech trees in New York have been in decline from beech bark disease since the 1960s, and now beech leaf disease is the latest threat. It can kill mature trees in 6 to 10 years and younger trees in just a few years. And loss of beech would cause changes in forest structure that would affect the whole ecosystem. Next. Beech leaf disease in North America. In North America, beech leaf disease was first found in Ohio in 2012, and it has been observed spreading outward from there since. In New York, beech leaf disease has been found on American beech, European beech, and Oriental beech. Next. In New York, DEC Forest Health first started hearing reports of striping on leaves and thinning beech canopies in 2017. We started tracking it as beech leaf disease the following year in 2018. At that time, the only detections were in the most southwestern part of the state, but each year since, we've seen a significant expansion of the disease range. And you can see here that we've already found 12 counties just this year. And just this week, it was unfortunately found in the Adirondacks. We get a significant amount of IMAP reports from Westchester and Suffolk counties where it is widespread throughout. It would be really great if, like Mitch was saying, to fill in the gaps, if we were able to get more reports in other areas where we don't know where it is. A lot of these new pinkish purple new counties this year, we've only found like 1 or 2 new sites. So it's not spread throughout, but it is spread throughout downstate and also in Western New York, unfortunately. Next. Go back one. That one, yeah. Okay, since beech leaf disease has only recently been recognized, its biology and vectors are not well understood. There is still much unknown and research is ongoing. Research by CARTA found that the foliar nematode Lookalikes crenadii mccannii causes beech leaf disease symptoms, but it is unknown whether the nematode causes all of the damage or whether it associates with a pathogen such as a virus or bacteria or fungus. The cause of its spread is also unknown. Birds and or human spread are 2 theories. So this Lookalikes crenadii, this is what beech leaf disease is called in IMAP. So when you are making IMAP submissions, this is what you would look up. You could look up beech leaf disease or Lookalikes. Next. Okay, so now I am going to go over what you need to know to survey. You could really survey all year. The striping is present on the leaves from leaf out, and it stays on the leaves after they even fall off. But the ideal survey time is late May through October, because that's when it leaves out, and then beech trees lose their leaves late, but eventually they do fall off. But so here you can see the banding at leaf out and then again in the fall. Next. The importance of pictures. This is the important part. I need good, clear photos of the banding in order to confirm reports. Neither of these photos show me clear evidence of beech leaf disease, and I would have to mark it insufficient to confirm because it's not giving me enough evidence here. Photos should be taken upwards through the light, showing the dark banding. Also, I just want to note, please only submit 1 point per area. We don't need individual trees submitted. If you're walking down a really long trail and end up far from where you started, maybe you could submit a point at the beginning and the end if you really wanted to, but overall 1 point per area is sufficient. Next. Identifying beech. Okay. So the first thing you'll need to know is how to identify beech. Beech trees naturally have smooth gray bark, though they often have beech bark disease, which makes the bark bumpy, and in public areas, they're often frequently carved into. Next. Beech leaves are simple and ovate in shape, and they have teeth. You can see here how each vein ends in a tooth, and the leaves are not hairy or sandpapery. Next. Here you can see a comparison between Eastern hopworm and American beech. You can see how on the Eastern hopworm, each vein does not end in a tooth. There are many more teeth on that one. Next. Symptoms of beech leaf disease generally progress from the bottom of the canopy upward, but can be found randomly throughout. Likewise, it is found in the understory before the overstory. Early symptoms include the darkened striping between leaf veins. Later, heavy banding can cover most of the leaf surface. This will impact the tree's ability to photosynthesize. After a few years of being infested, leaves will become deformed, chlorotic, and have a thickened, leathery texture. Next. Beech leaf disease symptoms are variable, so I'm now going to show you several more examples of the symptoms as well as other beech problem lookalikes that you could encounter. Here you can see some examples of light to moderate banding. The dark striping on the leaves is most visible looking upward through the light. Next. Here are a few more good, clear photos of the dark banding. Next. Yeah, there we go. And here is a good example of why you need to hold the leaves up to the light. Here in this first photo, these are going to be the same leaves. This first photo was taken looking down, and you can see how it's unclear whether beech leaf disease is present. Next. Then the same leaves shown up through the light, and the dark banding is clearly visible. So yeah, you can see here, these are the same leaves, and it's really unclear in the first one. Like I would say, like if I saw this first photo, I would say, yes, that looks really suspicious, but I'm going to need to see more evidence in order to confirm. But then in the second photo, it's very clear, and I would say definitely beech leaf disease. Next. Here's a little practice to spot the beech leaf disease symptoms in the photo. See if you can pick them out. Next. See, you can, that's where they are in that one. Next. Next. Yep. And then next. Next. So you can see, like sometimes I just get a photo like looking up through the canopy and like this, and that's fine. But sometimes it's harder to identify it to see where it is than others, especially in the lighter infestations. Okay, next. Okay, heavy symptoms. Heavily infested leaves are shrunken, curled, and leathery in texture. Next. Symptoms progress to puckering, curling, chlorosis, and necrosis. Next. At the latest stages, leaves will be deformed, leathery, and possibly shrunken. Okay, next. Okay, now I'm going to go over some other problems that you might encounter on beech, including erinium patches, aphid damage, and anthracnose. Symptoms are also variable on these issues and can look very similar to beech leaf disease in some cases. Remember, it's always better to report even when unsure and make sure to get good, clear photos. Next. The first lookalike that might be confused with beech leaf disease is the erinium patch caused by Eriophid mites. It is similar to beech leaf disease in that the damage is a gall, which is the tree's response to the organism. But generally, you'll see spots rather than stripes in general. And now there are two types of Eriophid mites that can attack the beech. And basically, it's just that one will attack the bottom of the leaf and the other one will attack the top. So, but basically, you'll see these spots. Next. For the erinium patch, there will be a felt-like area on the surface of the leaf, and there might be some puckering on the other side. Most of the time I see light spotting like this, but in severe cases, there can be some light striping. Beech leaf disease doesn't have that felt-like patch, and the thickened area takes up the entirety between the veins. Yes. The next lookalike is aphid damage. Aphids will cause a lighter banding versus the dark banding on beech leaf disease. Next. You can see here how the aphid curls are a much tighter curl on that part of the leaf, whereas beech leaf disease curls are an overall distortion of the whole leaf curling. Also, if you look at the back of the aphid damaged leaf, you might see old aphid casings. You can see in this last photo of beech leaf disease how difficult it can be to identify from the top down. The chlorosis could be confused with aphid stippling. Next. The final lookalike we're going over today is anthracnose, which on beech leaves looks like patches of necrotic tissue, usually on the end with the tip, but it could be towards the side as well. Next. And here is anthracnose versus beech leaf disease. Beech leaf disease necrotic tissue can be areas along the side or striping. Next. Management and prevention. Treatments are being researched, but unfortunately nothing has been approved for usage yet. DEC Forest Health staff are cleaning their boots with a boot brush and a 10% bleach solution in an attempt to prevent spread. If you are using a bleach solution, it is important to add new bleach every 24 hours because it loses its strength. Next. So that's it. Here's the sources I've cited and taken photos from. Next. Yep. For more information, please visit our beech leaf disease webpage, which you can find by Googling NYSDECBLD or by following this link. On that page, we made a beech leaf disease flyer, so you can look at that for more information. You can print it out to hand out to people. And also, if you have any questions about tree problems at all, you can contact the DEC Forest Health Information Line. I'm going to see if there's any questions in the chat.
Awesome. Thanks so much, Maria. I think there's time to maybe answer one question, and then other ones could be answered in the chat box.
Okay. It does not, it affect, so far we've seen it affect the different species of beech trees the same way. And we haven't, those are the species of beech that we found it on, and we haven't found it to not attack any types of beech yet. And does beech leaf disease present itself on all foliage of the tree? Oh, and I see that Jason has answered that. Yeah, it usually starts by just a little, like a few leaves, and then progresses from there. And it usually starts from the bottom upward.
Cool. Thank you.
And if you can also email me directly, I can put my email into the chat if anyone has any questions that you think of after. You can just email me and I can answer them later.
Thank you so much, Maria. That was great.
Thank you.
And if any other questions pop up, feel free to enter those into the chat. But now we will switch to jumping worm, and we'll hear from Annis.
Great, thanks, Mitch. While you're giving me screen control, I'll just say a couple of things. One, I'm so grateful that you guys are participating in this. I'm setting up a new experiment right now as we speak. I'm zooming in from a field office of the Audubon in Vermont. I'm very grateful for them to give me a desk. But the reason, when I start every project, the first thing I do is I look at iMaps and I look at the distribution of the species that I want to work on, and having that database to pull from for regional experiments is so invaluable. And instead of it taking 5 years to set up a project driving from place to place, we have this great database. So I'm always grateful for this, and I use it every single time. So thank you all for being here. Okay, if I did that, then I think we're good to go. And I do have to zip off pretty quickly after this, but I'm also going to put my email address into the chat box if you have questions that come up after. So quick overview of earthworms in North America. For the most part throughout New York State, the forests have established in the absence of earthworms, not just jumping worms, but earthworms in general. We do have a few native species, but they're pretty rare. They tend to be in coarse woody debris or in wet, very, very wet areas. So what you're seeing out there is, for the most part, either European or more recently jumping worms. So historical New York forest looks like this cartoon here on the left, where you have a lot of leaf litter falling in from the trees and creating a thick duff layer on the top of the soil. Those are the conditions that a lot of our plants and animals evolved into. So less of these leaves are being used up than what's being built up. When earthworms come into a system, you get something more like this picture on the right, where everything's very mixed up. And with jumping worms specifically, if any of you have seen that, what this means is that the top 6 inches, even 12 inches, can just be these very, very thick layer of castings that are pretty homogeneous all the way down. And you lose a lot of the diversity that you have in the understory. So the second wave of invasions, the first wave being the lumbricid earthworms coming over from Europe, are jumping worms that go by many different names, but I think jumping worms is the one that I kind of like. It's very descriptive. And we've got 5 species invading forests in North America, but it's really 3 in New York State that all look pretty similar for the most part. So on IMAP, when you do it, it'll just say jumping worms. We're not asking you to try and get down to species. That's a pretty tricky thing. So the first thing to look for and to take a picture of, I can often identify, confirm an invasion just based on the soil photo. So it's always good to include soil as much as possible. So we're looking for this really textured, either coffee ground type. If it's dry, it'll look a little bit like that. If it's wet, maybe it looks a little bit more like ground beef. But it's really distinctive, crumbly pieces of soil. You'll see a lot of erosion, maybe exposed mineral soil a little bit later in the year. And this is kind of what the chunks look like here on the right. What this means for roots is that it's very hard to grow. They dry out really quickly. It's tough for seeds to germinate under these conditions. And it's pretty unstable, so things can get pulled out easily by a deer taking a bite instead of clipping it off a plant. It'll kind of pull the whole thing out. And we do find that having these 2 things in combination, jumping worms and white-tailed deer, have more than just the sum of their parts in impact. So big impacts to places like golf courses, landscaping, parks, a whole food web of impacts that happen when you change this baseline community in forests. So it's really, really important. And as Maria mentioned, we don't have time to get into all of that, but you can ask me more if you want to later. So the first thing, as I mentioned, is looking for that granulated soil. Here's another up close picture of it here on the right. This is what jumping worm invaded soil looks like when it's a pretty strong invasion. The European lumbricid invaded soils tend to look a little bit more homogeneous, kind of like garden soil. And then here, if it's not invaded by any kind of earthworm, you'll tend to see lots of organic matter and leaves from many previous years building up. So right now we're here where they're starting to be juveniles into adults. So for context, where I live in Connecticut, they're all adults right now. And where I am here in Vermont, they're all juveniles. So if you're somewhere between, if you're in the Adirondacks, you're probably looking for juveniles right now. If you're a bit further south, you're probably looking for adults. And then if you were looking later in the year, you'd have to look for cocoons. But hopefully you'll have logged your invasions before then. All right, so first you're going to be looking for the soil. That's the first step. And you can do that pretty quickly. You can just scoop the leaf litter off from your shoe and look to see what the soil looks like. And then at this time of year, you can start looking for the worms themselves. So the first thing that's very noticeable is changes in their behavior. And I don't know if this is something I can do. Oh, it is something I can do. Look at that. So they wriggle pretty quickly. They kind of move like snakes, as you see some of those jumping out of the dish. Very distinctive, especially at first when you first move them, but then they kind of get tired out and they don't do a whole lot of jumping after the first couple of jumps. This is kind of a lazy, not super stressed out lumbricid earthworm, but these are the ones that are not the jumping worm. So they kind of pull themselves along by expanding and contracting as they go. The movement is a really good key to look for. As far as the actual worm itself, this is what a mature one looks like with the clitellum right here. They can be, so if you're in Vermont, you're looking for something about 2 centimeters. If you're further south, they're quite a bit bigger than that by now and getting bigger and bigger throughout the year. They're quite darkly pigmented, and in the light, they look very metallic or iridescent, quite distinctive. If you do have one big worm and you're trying to decide if it's a jumping worm or a European worm, there's a few other things you can look for. So the clitellum is the first thing to look for. That tells you if it's an adult or not. So on the jumping worms, it's going to be kind of like a ring that goes sort of all around the body of the worm, versus in the lumbricid worm, it's more like a saddle where it's just on the very back of the worm, whereas the front does not have the clitellum all the way around. The color is the other thing to look for. So in jumping worms, it's sort of white, pearlescent, or milky white, or mother of pearl, one of those colors. Can even be sort of pinky pearly or gray pearly, whereas with the lumbricids, it's tending to be brownish red, those kinds of colors. The other thing is the position of the clitellum. So here you can see on the jumping worm, it's very close to the head of the worm, versus in the lumbricids, it's closer to the middle of the body. If you do have a hand lens and you're looking, sometimes if your eyes are really good, you can see this just with your naked eye, but it is a little bit tricky. So microscope or hand lens can be helpful for this. Is you can look for the setae. This you can do even if you have a juvenile worm. So in the lumbricids, they tend to be paired setae. So that's going around here along the edge of the worm. This is a picture looking down the barrel of the worm of what it would look like. Versus on jumping worms, it goes all the way around. It kind of looks like bristles. This, if you're really a keen natural historian, you want to get it to species, this is a key that you can use for the ones that we have that are the most common in New York. I think this is recorded, so you can go back and look at it. I'm not going to walk you all the way through it. It's also not, it's not fully resolved. In a lot of cases, it can be tricky to figure out between all of them, and you have to use molecular methods to get at the species. So for the most part, you can just get down to just that it's a jumping worm for our purposes. So you can upload that presence or not detected records to IMAP. When I do my research, I often will have like a paired invaded, uninvaded site. So I use that not detected data just as much as the detected data. So multiple images for confirmation is great. You can just submit one. You can then dispose any of those worms by freezing or putting in ethanol. Those are sort of on the continuum towards the least painful way to euthanize the worms. And then if there's any additional observations, you can definitely put that in the comment section. I think they have that for these guys. Not having a ton of worms in the photo, just sort of one or two, something like that. Castings or some of the soil is good to include. Mature specimens are better than immature if you can get them. Of course, good resolution. Something for scale is helpful, like a ruler is the best thing, but if you don't have a ruler, you know, a coin or something standard sized is very helpful. Straight worm is better than a coiled up worm. And like I said, the fewer, the better. 1 or 2 is better than 20. So just really quick summary of what you should look for. So the soil, look for the granular appearance, look for the thrashing behavior, look for the bristle-like setae in a ring around each segment. And starting about now, when they're adults, you can look for the ring-like clitellum close to the nose of the body. And this is really important. We don't, similar to beech leaf disease, don't have any management solutions for treating the invasion once it's established. So putting efforts into not moving soil and especially mulch and plants around is key to prevent the invasion. Right. What do we think? One question or just push on?
If you have time, you could maybe answer one out loud or type it into the chat, whatever is most convenient for you.
Okay, let's see. So we'll start here. When reporting jumping worms, what should we say if there's granulated soil but no jumping worms visible? So if you just take a picture of the granulated soil and it looks distinctive enough like granulated soil, maybe hold up a little clump of it, that I mean, I'll talk to the IMAC folks to make sure that that's okay, that we don't actually see the organism. But for my purposes, I definitely know that I would call that invaded.
Yeah.
Yeah. Yeah. Yeah. Yeah. Okay. Great. So I will I'll take a look at the other questions and type anything else into the chat. Thanks a lot, everyone. I appreciate your efforts.
Thank you so much. And so now we've learned about beech leaf disease and jumping worm, and so I'll do a quick run through of the aquatics. So we have 3 aquatic plants included this year, kind of lumped together for the prizes and trophies. Water chestnut, which we've had for a while. That might have been the first species or the second species. We added Frogbit 2 years ago, and then this year our new species is Watercress. And so they're lumped together. Of course, they're all different, but a lot of aquatic plants do have some impacts in common. So some of the common impacts you'd see within an aquatic plant are that they outcompete native species. They form these dense monoculture mats, which then shades out the species that would normally be growing under the water. They can also alter water flow. They can kind of block it. And so on the right, there's a picture of what a European frogbit infestation looks like. And you can imagine that that could also impede recreational activities. So boating, swimming, paddling, fishing, that can all be affected by these dense mats of monocultures of aquatic invasive plants. And then here's another example of a really extensive infestation of water chestnut, and you can imagine how hard it would be to kayak through that, for example. And I'll just do a really quick one slider for water chestnut and frogbit, and I'm going to focus most of my time on the watercress because that's the newest one, and I think it's also maybe the most challenging species we've added so far. But for water chestnut, it's very distinctive. There isn't really much else that looks quite like it. It forms these rosettes. You can see in this bottom picture, that's what it looks like floating on the surface, these circles of leaves. And so these leaves are sort of like a diamond shape. Some people say triangle because it's kind of flatter on the end, but it's sort of like this diamond shape. And then there's these teeth all along the edge. You can see it in this photo of a close-up of a young plant. So they can be small like this in the early stages. And I don't know if you'll be able to see in any of these pictures, but the stem attached to the leaf, that's called the petiole. It's actually inflated a little bit with air, and that's why these leaves lay flat on the top of the water. Yeah, so that's the leaves. So that's the thing you're most likely to see because it'll be floating on top of the water. These are rooted plants, and their submerged stem can grow up to 16 feet. So in theory, it could be in water that deep, but kind of the more common place you'll see it will be towards the shoreline and in the shallower areas. And some of you might have encountered its fruit before. It's called a nutlet. And it's very spiky. It has these spines. They're very painful if you step on them. These are, they each produce a single seed. And so water chestnut can grow from these if you see these. A lot of times you'll see the spent ones that are no longer alive washing up on shore. And they do have a flower, which you might see this time of year. They're small white flowers. So that's water chestnut. Frogbit is another floating aquatic plant. It looks a little bit different though. So it has these small rounded heart-shaped leaves. They kind of look leathery when they're in these mats like this. And they're actually often free floating. So the roots won't normally be like embedded in the substrate like water chestnut would be. And it also sends out stolons to like kind of spread sideways, which allows it to form dense mats. And for the flowers, you can see these little white three-petaled flowers. And it's actually a dioecious plant, which means there are separate male and female plants. And I believe in New York, we only have the female plants so far. But they have these small white flowers. And to get a sense for the size, you can see it in the hand of this iMap observer. So there are other plants that kind of have that rounded heart-shaped leaf that you'll see floating in the water, but they're very large. So Frogbit looks very different because they're so small. So that's Frogbit, and then I'll spend more time talking about watercress, Nasturtium officinale. So this is kind of a very good example of an underreported species. So in the map, we have orange towns or towns shaded in orange where there's a watercress report. And it looks very different from what we usually expect with an invasive species. Usually we see kind of like a cluster of positive records and then sort of a leading edge and then not much beyond the leading edge. With this, it's just kind of like scattered all throughout, but a lot of blank spaces in between where we're seeing it pop up. So it could be a lot more if it's, for example, if it's all the way out here and it's all the way down here, it really could be in many places in between. And so this species is native to Eurasia. It also likes shallow water and wet areas, so lakes, ponds, streams, muddy banks, the same sorts of places you'll see frogbit or water chestnut. But this watercress, it also can be found in drainage ditches and puddles and springs, which you wouldn't necessarily expect frogbit or water chestnut in a puddle, for example, but watercress, you might find it there. This is the same watercress that you can eat that you might find at the grocery store. So that might be part of how it's spread, so escaping cultivation or from discarded food scraps. But it forms these dense mats, crowd out native species, alter flow, etc. And so in this picture, you can see in the foreground, watercress has kind of spread. They're kind of a low plant at the surface of the water. It's kind of spread into this mat, which can cause issues. For identification, things to look out for. These are what the flowers look like. They're small, white, four-petaled flowers, and then those develop into these bean-like fruits, which are called calyx. And there are a lot of plants that look like that. Like you might be familiar with garlic mustard, which is another invasive. There's a lot of other species in this same family that look very similar, but most of them don't really occur in the water like watercress does. So that's one thing to keep your eyes peeled for when this sort of plant is growing in water. And the leaves, so what I'm putting my mouse around right now, that's one leaf, and then the little green circles are the leaflets. So they have 3 to 9 leaflets. This one up here has 5 leaflets. They kind of have like wavy edges, and the one at the end is kind of rounded. So this leaf shape is really the most distinctive thing to look for with this species. And you can also see what it looks like with a couple plants kind of poking through the leaves, floating on top of some water. And then it has the slender fruit pods with seeds arranged in 2 rows. The stems and leaves are hairless, and the stems from the root to the nodes up to the leaves are hollow. There are a couple plants that look like it that you might find in the water. So I think the most common one that could be confused would be Pennsylvania bittercress, which is a native species here. So at first glance, it does look pretty similar. The flowers are kind of the same. One difference is that the fruits are very long. They're like very long and narrow, which looks a little bit different than the watercress. And also the leaves are a little bit different. So they have kind of a similar structure, but the leaflets are very long and narrow. So you can see, especially near the top, they're very long and thin as opposed to the rounded look with watercress. And also they can have a lot more leaflets. So watercress is in the 3 to 9 range, but on this Pennsylvania bittercress, you can see there can be much more. So this one on the bottom here has 2, 4, 6, 8, 10. I'm seeing maybe 11 leaflets. So that's how you know that that's not watercress. So that's probably the most common one that would cause confusion. One other one that looks kind of like it is actually on our rare species list. So lake watercress, Roripa aquatica. This is, so you can see the flowers look kind of similar, but the upper leaves look quite different. So they're kind of long and narrow with these teeth. And then the lower leaves below the surface, they do have they're like kind of separate into those leaflets, just like watercress, but you can see they're way more finely separated. So they're kind of spiky looking. They look almost like carrot leaves. So that's a very distinctive from the watercress. And that's a rare species. I think it's classified as imperiled in New York State. So if you find that, that's cool too. Feel free to reach out to us and we can connect you with someone to report that finding. But really the thing you want to look for are these distinctive leaves with the 3 to 9 leaflets and a rounded one at the top. And one thing that's very important to get a picture of, if it's there, is the fruit. So Nasturtium officinale watercress has these 2 rows of seeds. This is a very close-up picture from a scientific study, but there is actually another invasive species called one-rowed yellowcress. It's a species of the same genus, and it has 1 row of seeds. So they're closely related, and they look very similar. The only way to distinguish them is the fruit. So I encourage you, if you find watercress, report it as watercress, Nasturtium officinale, but try to get a good picture of the fruit. And we might be able to tell if by chance you've found this other more rare species. And I should mention, you're much more likely to find the normal watercress. This one-rowed yellowcress is not really known in most of the state, so you're not likely to find that one. This is just kind of a heads up that it's possible that when we have people out there looking for watercress, they might come across this other species. And so that's our 3 aquatics. And so, yeah, those are great to look for whenever you're out on a water body. They like the slow flowing or even stagnant water bodies, often shallower portions. So that's the aquatics. And since we're running short on time, I'll do my quick IMAP overview before looking at the chat box. So just so people know how IMAP works, records go in as unconfirmed, so anyone can submit a record. And then those are then reviewed by some of our confirmers. And so it's really important to have a nice photo where you can very clearly see the identifying features. For example, this tree of heaven photo, you can see those rounded teeth at the base of the leaves. So that makes it very easy to confirm. And then those records become part of the official distribution for that species. So email alerts will go out. They'll be part of the official range map for the state. They feed into regional analyses, which influence management strategies and more. And so the first thing to do to report to IMAP is you need to make sure you have an account. So you go to our homepage at nyimapinvasives.org and click login. And if you have an account, you can log in or reset your password. If you don't have an account, you would create it and make sure you follow the text that pops up to go check your email and activate your account, because you'll need to activate your account before you can use it. And once your account's set up, the app is kind of this 3-step process where you set up your app. So you put in your account information and tap retrieve IMAP lists. And you have to do that while you're connected to the internet, and it will kind of connect your account to your app. And once that's all set, then you can go out and check for all 6 of those species, and record observations while you're out in the field, and it will capture the location from your phone. And you can see you can select between species detected and species not detected. So if you go out and check for these species and you don't find them, it's really useful to have that documented, to know that you went out and didn't look for it and didn't find it. And then, so say you go out and you record 10 records throughout the day. Like Maria said, we don't necessarily need you to stop at every individual plant and report a record each time. One general rule of thumb could be you don't necessarily need to record multiple observations within 100 meters. So if you're going down a trail or something, kind of visualize like a football field, and you don't need to report in between that length interval, kind of space them apart. And then once you get back into connectivity, then you just upload those records. So they leave your phone and then go onto the online database. And just quick take-home messages. Hopefully you've learned a few species to keep an eye out for. And so I encourage everyone to go out throughout the month of July and check for those species and report presence or not detected to IMAP. If you report presence, that photo is super, super important for confirming. We'll actually have a blog coming out a blog post next week coming out about how to take good photos for confirming. So keep an eye out for that. And also I encourage you all to report throughout the month of July, and maybe you'll be in the running for a prize, or at least you'll help your prism get a trophy. And just as a tip, if you report fake species from your mobile app, you can do that before you go out. And that kind of lets you test out the app and make sure you know how to use it before you get out in the field to actually report the invasives. And I won't take the time to show this right now, but you can kind of find all the information you need for the challenge on this nyimapinvasives.org/challenge page. So there's information on how to identify the different species. I'll be adding Watercress to that webpage soon. And you can check out the leaderboard. So there's a leaderboard that you can click out to, and you can click on the individual leaderboard or the prism leaderboard and see who the top reporters are so far for confirmed, and then also for not detected. And at the end, we'll add up the confirmed and the not detected to choose our final winners. So thank you all so much for joining, and thank you so much to our presenters. I will do a formal end to this and stop the recording, and then we can go through any questions.